Good morning to one and all. I'm here to talk about the spontaneous generation and the germ theory. So, what do you mean by spontaneous generation? A spontaneous generation is the theory that states that all living organisms could arise from the non living matter under the right conditions. This idea, also known as abiogenesis or heterogenesis, and was widely accepted for centuries and suggested that things like maggots could appear from rotting meat or mice from cheese and bread. While now discredited, it was a cornerstone for early biological thoughts. This theory was widely accepted for over 2000 years, influencing scientific thoughts until the 17th and the 18th century. The main scientist who is behind the theory of spontaneous generation was Aristotle. Aristotle believed that living organism could originate from non-living substances provided the material contained vital heat or pneuma, which he believed to be the life force. He observed that frogs appear in newly flooded areas and maggots seem to emerge from decaying meat, leading him to conclude that the organisms arise spontaneously. Francisco Redi was the second scientist involved in the spontaneous generation debate. Francisco Redi's experiment in 1668 disproved the theory of spontaneous generation by demonstrating that maggots did not arise from rotting meat itself but from fly eggs laid on the meat. He showed that if flies were prevented from reaching the meat, no maggots appeared. Reddy's experiment involved placing meat in jars, some of which were sealed, covered with the gauze, and some left open. He observed that maggots only appeared in the open jars, where the flies could lay and land eggs. And on the gauze of the gauze covered jars, but not in the sealed jars. This indicated that maggots were a result of fly eggs, not spontaneous generation from the meat. Reddy's work is considered a key early example of the scientific method using controlled experiments to test a hypothesis and refute a long held belief. So, we can see his experiment in this picture. In this picture, we can see that he took three jars and he put the meat, raw meat, in each of the three jars. The first jar was an open jar. It was not covered in any ways. The second jar was a cork sealed container, and the third jar was covered with a gauze. So, and after some times, he saw that the open container had the growth of maggots and flies. The second container, which was a coxal container, there were no formation of maggots in the meat. But in the third one, we saw that in the surface of the gauze there were maggots and the flies lying. So, thus, he understood that the spontaneous generation theory was not correct. The third scientist involved in the spontaneous generation theory debate is John Needham. John Needham's experiment in 1745 supported the theory of spontaneous generation by demonstrating the appearance of microorganisms in boiled broths that were then sealed. He believed that boiling the broth would kill any existing microorganism, and the subsequent appearance of the new ones indicated that life arose from non living matter. However, his experiment was later criticized for not boiling the broth sufficiently to kill all microorganisms, and his sealing method was not completely efficient. So, if he if he wanted to he wanted to prove that the spontaneous generation theory was correct. So what he did was that he took a flask, he filled the flask with the broth, and he heated it, and he left for it to cool, and then he covered the flask. It is not shown in this picture, but he actually covers the flask after he let it to cool. So, what happens is that the air Air in the atmosphere also already entered into the flask containing the broth when he allowed it to cool before sealing the jar close. And after giving it some incubation time, what he observed that there was the growth of microorganism. Even though he told that this proves the spontaneous generation theory, later we come to know that his sealing methods were not correct. If he had sealed during boiling, it would not the microorganisms would not have been grown, but he didn't thought it like that, and he said that the spontaneous generation theory was correct. The next scientist was Lazaro Spallanzani. He said that involving boiling broth in flask and then sealing them was wrong from the John Needham's part. So what he does that he observed that no microorganism grown in the sealed flask while he sealed and boiled the flask at the same time. Lazaro Spallanzani specifically targeted the work of John Needham, who had previously conducted experiments that he interpreted as supporting the spontaneous generation theory. So, Lazaro Spallanzani's experiment says that when he took a flask containing a broth, and obviously the unsterilized broth may contain microorganisms, he subjected it to heat while closing the flask. After some time, he let it cool and give it some incubation time. And what he observed that there was no cells growing, unlike the John Needham's experiment. So whatever he said, criticizing the John Needham's experiment was true, and also it helped in disproving the spontaneous generation theory. And the final disproving of the spontaneous generation theory was done by the Louis Pasteur. Louis Pasteur's experiment disproved the theory of spontaneous generation by demonstrating that microorganisms in the air contaminate sterile broth, not the air itself. He used swan neck flask which allowed air to enter but trapped dust and microbes, preventing them from reaching the broth. This experiment supported the germ theory of disease and helped to the advancement in sterilization and pasteurization methods. So, what he did was that he took two swan neck flasks. He filled it with water or a broth. And then what he did was that he he broke one of the swan neck flask neck, and after heating it, he let it cool for some times and give the microorganism some incubation time. And what he observed was that the unbroken swan neck flask didn't show any growth of microorganism. Why? Because he also observed that the curves of the swan neck flask trap the air and the microorganism, thus preventing the unsterilized and the contaminated air from contaminating the broth, and uh, thus giving strength to his disproval. In the second, the broken swan neck flask, we see that the microorganism grow and the broth is contaminated. That because uh, after the neck is broken, it leaves space for the contaminated air to fill inside the broth and contaminate it. The next person, who also supported Pasteur in disproving the spontaneous generation theory was John Tyndall. Tyndall's experiments, particularly his use of Tyndall chamber, a specially designed box to filter air, showed that sterile conditions could be maintained if air was free of particulate matter. He also demonstrated that heat resistant endospore could be present, which he explained why some previously sterilized methods might still show microbial growth. Tyndall's research also led to the development of Tyndallization, a process of sterilization involving repeated heat and cooling to eliminate heat resistant microbes. As we see the Tyndallization method, what he does is that he boil in 100 degrees Celsius, boil a broth in 100 degrees Celsius, and turns out it gets sterile, but still there will be growth of some little percentage of microorganisms. So what he does is that instead of letting it to cool down, he again boil it in a while leaving a very little period in between both the heating. What result he got was that the microbial load in the boiled broth was very low. Next is the germ theory. The germ theory states that certain diseases are caused by the invasion of the body by microorganisms. These microorganisms are too small to be seen with the microscope. Therefore, the germ theory, the miasma theory, was widely accepted. This theory proposed that diseases were caused by poisonous air emitted from the decaying matter. Louis Pasteur, a French scientist, conducted experiments that challenged the idea of spontaneous generation and supported the germ theory. According to a biology resource, he showed that microorganisms are not created by decaying matter, but rather came from the environment and can cause fermentation and spoilage. His work with swan neck flowers demonstrated that microorganisms in the air were responsible for contamination. Koch postulates. The Koch postulates also added to the germ theory, giving it more foundation and strength. The Koch postulate states that the microorganisms must be found in all cases of disease, but not in a healthy individual. The microorganism must be isolated from a deceased individual and grown in pure culture. The isolated microorganism should cause the same disease when introduced into a healthy, susceptible individual. The microorganism must be reisolated from the experimentally infected individual and shown to be the same as the originally isolated microorganism. So, hope you understand what I've explained, and thank you so much.